Hello students, welcome to EPG Pathshala. I am Anil Kumar Sharma from Department of Pharmaceutics, Delhi Institute of Pharmaceutical Science and Research, affiliated to University of Delhi. Today, I am going to talk about module Nanoerythrosomes, Preparation and Characterization from paper Novel Drug Delivery System 2. Learning objective of today's module are: first, preparations of nanoerythrosomes; second, characterization of nanoerythrosome; third, kinetics of drug release; fourth, in vivo behavior; and lastly, applications of nanoerythrosomes. Preparations of nanoerythrosomes involve formation of erythrocyte ghosts, hypotonic osmotic lysis method that again consists of three parts. First one is dilutional hemolysis, second is press-well dilution, third one is dialysis. We go to a dilutional hemolysis. Dilutional hemolysis involves dilution of the RBCs in a hypotonic buffer with the drug to be encapsulated up to 2 to 20 volumes. Then these are resealed using isotonic buffer. Similarly, we use press-well dilution method, but in this technique the cells are not lysed. These are not lysed ruptured, but they are again resealed in isotonic buffer solution. Lastly, the dialysis technique. In this technique, the drug as well as your drug to be encapsulated, both are kept in isotonic buffer, and this bubble, the dialysis sac, is flattened using air bubble. And this is again kept in a lysis buffer, that is around 200 ml of the volume. Then this is taken again ruptured, and after centrifugation the cells are resealed. Dilutional hemolysis. The swelling and lysis of RBCs upon exposure to hypotonic medium, that is 0.4% weight by volume sodium chloride, may encapsulate a number of drug molecules. One volume of wash cell might be treated with 2 to 20 volumes of substances to be encapsulated within the RBCs using a hypotonic buffer at 0 degrees Celsius for 5 minutes. The resealing is carried out in isotonic medium at 25 degrees Celsius. Next method is press-well dilution method. In this method, regulated swelling of erythrocytes without rupturing by exposing them in a moderately hypotonic solution, that is 0.6% weight by volume sodium chloride, succeeded by centrifugation at low G scores leading to lysis of cell membrane, facilitate entrapment of active pharmaceutical ingredient. The resealing in isotonic environment follows afterwards. Briefly, the hemolysis and resealing is executed in three phases. First, one volume of wash cell is dispersed in 5 volumes of moderately hypotonic buffer. Second, following placement at 0 degrees Celsius for 5 minutes, the erythrocytes were retrieved by mild centrifugation. Third and the last, the swelling process is further prolonged by incorporation of a volume of hypotonic buffer similar to half of the volume of the distended RBCs to cause lysis of the cells. The substance to be entrapped is present in hypotonic buffer medium, and the RBCs are permitted to stay lysed for 10 minutes at 0 degrees Celsius. The re-establishment of tonicity and resealing of the membrane follows afterwards. Last method is dialysis. The dialysis bag has a blend of washed RBCs of hematocrit value 85 to 95% and phosphate buffered saline. is submerged in 200 milliliters of lysis buffer and agitated for 2 hours at 4 degrees Celsius. The tonicity of the dialysis membrane was recovered by either substituting the adjacent medium with isotonic buffer at ambient temperature for 10 minutes, that is known as annealing, or capped in 200 milliliter of resealing buffer, that is hypertonic buffer, at room temperature for 30 minutes. The drug was incorporated by its dissolution in the isotonic cell dispersing buffer prior to agitation or by placing the drug within dialysis sac when rotation was over. The hemolysis and resealing is accomplished in three stages. First, washed RBCs of hematocrit value 85 to 90% are blended with phosphate buffer saline, having the drug to be encapsulated. The mix is afterwards kept in dialysis sac. The dialysis sac is distended using an air bubble and sealed in such a fashion that the cell dispersion engages internal volume not exceeding 75%. The packed dialysis sac is afterward capped in a bottle having a minimum of 200 ml of lysis buffer, that is 0.1% weight by volume sodium chloride at 0 degrees Celsius, and positioned over a mechanical shaker for 2 hours at 4 degrees Celsius. Preparations of nanoerythrosomes. First involve the preparations of erythrocyte ghost. After the preparation of erythrocyte ghost, the nanoerythrosome could be made by using first technique, that is extrusion, second technique, that is sonication, and the technique known as electroporation. Extrusion technique involves the extrusion of erythrocyte ghost from a membrane filters of different porosity to yield number of nanoerythrosomes. One RBC may yield around 4,000 to 6,000 nanoerythrosomes. The nanoerythrosomes so formed are tagged with uranyl acetate and visualized using microscope. Alternatively, the sonication is also a technique by using different sonicators. Number of sonicators are available such as probe sonicator or the bath sonicator is used to transform the ghost into the nanoerythrosomes. Alternatively, we can uh, break the ghost into tiny vesicles by using dismembrator. 2. Electroporation. This technique involves the use of external electric field to lyse the RBC. Under the influence of electric field, the RBCs are lysed and perforations are created. By the perforations, the drugs can be encapsulated, and this method is a good alternative to existing methods because it can encapsulate large molecules such as bovine serum, albumin as well as ribonucleases. Extrusion. The red cell's ghost dispersion of 50% hematocrit is passed across the 25 millimeter polycarbonate membrane filter of porosity of 0.5 micron meter. The nanoerythrosomes are resulted by 8 to 10 sequential extrusions under the influence of nitrogen pressure. The nanoerythrosomes so resulted are tagged with uranyl acetate and they are visualized using a microscope. The extrusions are carried out in a thermostatically regulated extrusion system at 37 degrees Celsius, and the ultimate formulation is placed in a refrigerator at 4 degrees Celsius. Sonication. Nanoerythrosomes may be made by sequential sonication by using either probe sonicator or the bath sonicator. An extrusion through a 200 nanometers pore size polycarbonate membrane. Alternatively, we can use a dismembrator for RBC's ghost to be transformed into tiny vesicles. These are known as nanoerythrosomes. Electroporation. This method is based on inducing pores in the erythrocyte membrane by exposing the erythrocyte to a strong external electric field. These pores were able to admit molecules of different size. This method of encapsulation is a good alternative to other commonly employed techniques. This procedure involves occurrence of electrically stimulated permeability alteration at extreme membrane potential divergences. The material may be encapsulated while application of an electric pulse beyond threshold voltage of 2 kV per centimeter for 20 microseconds. The desirable strength of electric field is 1 to 10 kW per centimeter and discharge duration is 20 to 160 microseconds. The potential differentiation transversely with reference to lamina is created either straightforwardly by inter or intracellular electrodes or in a roundabout way by affecting internal electric field onto the RBCs. The degree of aperture creation is governed by electric field power, pulse duration, and the ionic force of dispersion medium. By this method, large molecules like bovine serum albumin and ribonucleases may be encapsulated. These may be incorporated into the osmotically swollen and electrically pierced RBCs. The nanoerythrosomes of form may be characterized for various parameters involving physical characteristics, cellular properties, biological features, and physical characteristics involve size, shape, surface morphology, drug content, hemoglobin content, and the other parameters. Cellular properties involve their sedimentation, there are other things like shape or the turbidity or the deformability or the osmotic shock. These all are covered under cellular properties. Then comes the biological features. The RBCs, having the immense potential to be used as a drug delivery system, are also part of our biological system. They are biodegradable, biocompatible, and therefore the biological features are to be evaluated as well. Characterization of nanoerythrosomes. The nanoerythrosomes may be characterized for drug content. The deproteination of packed nanoerythrosomes is carried out using acetonitrile, and the vesicles are then centrifuged at 700 G for 10 minutes. The drug content is afterward determined by analyzing supernatant by using a spectrometer. Next parameter is in vitro drug and hemoglobin release. Empty as well as loaded nanoerythrosomes are capped at 37 degrees Celsius in phosphate buffer saline of pH 7.4 at 50% hematocrit in a metabolic rotating wheel incubator bath. At regular intervals, the aliquots are taken using a hypodermic syringe equipped with a 0.8 micrometer membrane filter. Percentage hemoglobin may accordingly be computed at several time points by using spectrophotometer. Besides, laser light scattering can be employed to determine hemoglobin content of a single red blood cell. Next parameter is osmotic fragility. It is a credible variant for in vitro assessment of carrier RBCs in terms of shelf life, in vivo endurance, and influence of entrapped materials. When erythrocytes are subjected to solutions of different tonicity values, their shape alters in order to maintain osmotic balance. To determine the impact of changing tonicities, the RBCs are subjected to saline solutions with diverse tonicity at 37 degrees Celsius for 10 minutes. The dispersion subsequent to centrifuging at 25 G for 15 minutes is analyzed for drug either or hemoglobin release. Next parameter is osmotic shock. This entails an abrupt introduction of drug carrying red cells to an ambience which is beyond isotonic. to assess the capability of resealed erythrocytes to bear the stress and keep their veracity and form. Placing the resealed erythrocytes in distilled water for 15 minutes succeeded by centrifugation at 1000 G for 15 minutes can lead to liberation of hemoglobin to diverse extent, which might be determined by using spectrophotometer. The other parameter is turbulence shock. This factor points towards the influence of shear force and pressure by which resealed red cells preparations are injected on the form of resealed erythrocyte. The packed RBCs are forced to pass across a 23 gauge hypodermic needle with rate of flow amounting to 10 ml per minute. The sampling of the dispersion is carried out subsequent to each passage and centrifuged at 300 G for 15 minutes, and hemoglobin content percolated is assessed by using spectrophotometer. Next parameter to evaluate is surface characteristic and percentage cell recovery. Phase contrast optical microscopy, transmission electron microscopy, and scanning electron microscopy are the microscopic techniques employed to characterize the shape, size, and surface attributes of packed RBCs. The cellular revival may be computed by determining the number of unbroken RBCs per cubic millimeter using hemocytometer. Erythrocyte sedimentation rate. It is an approximation of the dispersion stability of erythrocytes in plasma and pertains to the count and morphology of RBCs and relative content of plasma proteins, in particular fibrinogen and alpha beta globulins. This test is carried out by calculating the rate of sedimentation of erythrocytes in a standard tube. In general, value of ESR is 0 to 15 millimeter per hour in healthy humans. The ESR over normal range is signal of active but murky disease processes. Next parameter is deformability. It characterizes the effortlessness of journey of RBCs across the fine capillaries and reticuloendothelial system, hence assess the life duration of the cells. The deformability is evaluated by observation of time elapsed in course of certain volume of cells across capillary of 4 micron meter or polycarbonate filter having mean aperture size of 4.5 micron meter. This is performed to assess the viscosity profile of the RBCs and is based on viscoelasticity of the cell membranes, viscosity of the cellular components, and the surface to volume ratio of the cells. Viscosity and sedimentation behavior follows. The viscosity is measured with the help of Brookfield viscometer. Sedimentation volume of preparation is yielded by recording the height of sediment in a graduated measuring cylinder by using the formula F equals V ultimate upon V original, where F represents sedimentation volume, V0 is original volume of formulation, and Vu is ultimate volume of the sediment. Centrifugal stress. The nanoethrosome drug combinations are centrifuged at altering RPM using a cooling centrifuge at 4 degrees Celsius for 15 minutes. Afterwards, drug leaching in supernatant was analyzed. Next is hematological test. Regular clinical hematological diagnosis may also be performed for drug-loaded RBCs that covers average corpuscular volume, average corpuscular hemoglobin content, ESR, etc. Stability. Stability studies of the formulated nanoerythrosomes were performed by storage of all the formulations at 5 plus minus 3 degrees Celsius, 30 plus minus 2 degrees Celsius, at 65% relative humidity, and at ambient temperature for 2 weeks and 1 month duration. Next parameter is in vivo survival. A number of procedures are suggested to evaluate in vivo survival period, such as radio tagging of RBCs by 51 chromium or fluorescent marker like fluorescein isothiocyanate or encapsulation of 14 carbon sucrose or gentamicin. The endurance of tagged cell is determined by flow cytometry. From other than the tests which have been discussed earlier, there are more tests we can. The nanoerythrosomes can be characterized by cell sizes, lipid composition, membrane fluidity, density gradient separation, sterility testing, pyrogen testing, animal toxicity, because these RBCs have to undergo into the parenteral system. Kinetics of drug release from nanoerythrosomes involves the drug release from nanoerythrosomes obeys zero order kinetics. Zero order kinetics, there are number of reactions kinetics, but zero order, first order, pseudo zero order, pseudo first order, but the release order that nanoerythrosomes are found to follow is zero order. Zero order kinetics means a constant fraction of drug molecule is going into dissolution irrespective of its initial concentration. That means we can ensure the drug delivery for longer period of time. This facilitates our drug delivery for sustained period as well as to target the specific organs like liver, lymph, spleen, or the organs of reticuloendothelial system. This behavior has been observed for number of drugs. That means these are following zero order release kinetics has been observed for. Such behavior has been reported for artesunate nanoerythrosomes too. Now we will come to in vivo behavior of nanoerythrosomes. You see students, how your system is going inside the biological systems and how it is going to behave. That is called in vivo behavior, how it is going to survive there. So nanoerythrosomes offer prolonged circulation period. We have discussed sustained drug delivery via nanoerythrosomes. These nanoerythrosomes are offering you the drug you take once and forget the remaining dosage regimen because it is offering you sustained drug delivery. These vesicles, these nanoerythrosomes are preferentially or adsorbed onto the membranes of cells. Normally, while they are going to the system, they are adsorbing onto the membrane of cells and liberate the drug in the proximity of cells. That means most of the drug is residing in the vicinity of cells. That means most of the drug is available to the cell or to target. That means these are offering you the ability to target macrophages too, because these are actively taken up by the macrophages, and macrophages intake makes them a suitable candidate to target macrophages system as well as the reticuloendothelial system. Applications of nanoerythrosomes. Nanoerythrosomes have numerous potential uses in different regions of human and veterinary therapeutics. These cells might be employed as circulating vehicles to distribute a drug for long duration in circulation or in targeting selective organs like the liver, spleen, and lymph nodes. Most of the drug delivery investigations utilizing drug containing nanoerythrosomes are in the preclinical phase. Some clinical researches have yielded promising outcomes. The use of nanoerythrosomes are in following area: sustained drug release. These are used for prolonged delivery of antineoplastics, antiparasitics, veterinary antiamoebics, vitamins, steroids, antibiotics, cardiovascular drugs. The nanoerythrosomes may also be used for drug targeting. The RBCs with altered surface are employed to focus organs of mononuclear phagocytic system and reticuloendothelial system. Targeting reticuloendothelial system organs resealed erythrocytes by altering their surface attributes using antibodies, glutaraldehyde, sialic acid, sulfahydryl, and surface chemical cross-linking. For example, delivery of 125 iodine labeled carbonic anhydrase encapsulated in red cells chemically attached using sulfosuccinimidine propionate. Targeting the liver enzyme deficiency or replacement therapy. The enzyme used such as beta glucosidase, beta glucuronidase, beta galactosidase. If glucocerebrosides are deposited in the liver and spleen, these elements may be cured by glucocerebrosidase loaded erythrocytes. Treatment of hepatic tumors. The drug daunorubicin diffuses fastly from the cells while encapsulating, thus covalently linking with erythrocytic membrane using glutaraldehyde or sacchonitic acid as a chemical linker is fruitful. Treatment of parasitic diseases. Because of specific deformation of resealed red cells within reticuloendothelial system organs such as liver and spleen, confer them ability to be used as carrier for delivery of antimalarial, anti-leishmanial, anti-amoebic medicines. Removal of reticuloendothelial system iron overload. Desferoxamine carrying RBCs have been utilized to cure extra iron deposited owing to several transfusions to thalassaemic patients. Targeting organs other than those of reticuloendothelial system. The different strategies such as encapsulation of paramagnetic particles, photosensitive substances accompanied by the drug and antibody association to RBC's membrane for selectivity of activity. Students, now we will discuss some interesting facts about the nanoerythrosomes. First one, a technique or the procedure used to load drugs into erythrocytes or the red cells in stepwise fashion that utilizes instrument known as red cell loader. Red cell loader is the instrument which is loading the drug in a sequential manner. Second, some drugs also induce endocytosis, which enables encapsulation of drug molecules. Itself, this process of endocytosis is enabling the drug molecules to be entrapped into the RBCs. Next technique known as phase contrast microscopy we use for visualizing. Other techniques are also available, say transmission electron microscopy or the scanning electron microscopy, but the most common technique, that is phase contrast microscopy, is commonly employed to check the morphology of nanoerythrosome. It reveals the surface characteristic of your nanoerythrosomes. Next is, a wide variety of drug may be entrapped within the RBCs. The drugs possessing hydrophilic characters are the preferable drugs. They are the most compatible drugs which can be entrapped into RBC, but the drugs which are having hydrophobic characteristics are also entrapped into the drug. There are evidences of number of research papers are saying that this drug is hydrophobic, but still it can be entrapped into the RBCs. Next characteristic, the functional group, like groups, amino group, carboxyl group, thiol group, hydroxyl group. These are normally used for coupling to drug molecule. They are used as chemical linkers. So we will conclude whatever we have learnt in this module. Firstly, preparation methods. The preparation methods involve, first of all, making of RBC ghost by three methods. That methods were press well dilution or hypertonic lysis or the dialysis methods. Afterwards, the Ghosts were converted into nanoerythrosomes by three techniques known as extrusion, sonication, and the electroporation. Then, these were characterized for number of parameters. This included cellular as well as biological or the physical attributes of the nanoerythrosomes. Now there is the need to formulate nanoerythrosomes with lower immunogenic profile. That means there is no threat of immunogenic response to be triggered by use of such carriers. That is advantages for nanoerythrosomes as compared to other carriers like nanoparticles or the liposomes or the other synthetic carriers, because the RBCs are the cells or the carriers without toxicity, they are biocompatible, they are biodegradable. Now, this immunogenic profile can be reduced by coating with certain molecules like polyethylene glycol that will definitely lower the immunogenicity. Now the nanoerythrosomes coupled with antibodies. This is the test of diagnostic significance. So this can be increased test sensitivities. This is helping to increase the test sensitivity as well. And further, controlled and site-specific drug delivery is also possible by use of nanoerythrosomes because they are possessing all the essential characteristic of RBCs or the red cells. So this is providing zero order release. By virtue of that, this is offering control as well as site-specific drug delivery. Recently, a company known as DiagnoCure Incorporation has been granted a US patent for mitigating cancer's adverse effects. Thank you very much.